which allows more branching of the binding leaves. From figure 3, the mortality percentage difference of method 1 and method 2 is found to be 13.3%. Thus, this shows that the Parkia speciosa thrive better under the treatment of method 2. In conclusion, with a total sample size of 105 plants monitored over the period of 1 month, the subsequent plant growth is monitored and measured by comparing the average number of binding leaves, the average height, and also the mortality between the two methods. The results obtained fit our hypothesis that the introduction of the inoculation during the germination stage actually is more effective. The average growth in the direct addition of inoculation during the germination stage actually have a significantly higher growth as compared to those with the addition of inoculation to the soil. The rhizobia have established a symbiotic relationship with the plants during the germination stage, thus able to undergo more nitrogen fixation and also helps the plant in the growth. However, as the concentration of the inoculant increases, this will mean that the plant will have to provide more nutrients for the larger population of bacteria, thus it will compromise the growth of the plant. The mortality at the end of week 4 is also found to be at a low percentage of 7%. Thus, this will mean that there are a higher chance for these plants to survive when they are transferred to the degraded secondary forest. With these preliminary findings, we can do an extended project at Parkia Garden West, whereby the performance of the Parkia speciosa plant will be continued to be monitored by comparing the average height, the number of binding leaves, and also the mortality when transferred to the degraded secondary forest. The nitrogen fixation ability of these plants will